Yes, so I'm working in David Miller's lab on the molecular response of Acroporamylipora juveniles to ocean acidification, and I will show you today some preliminary results of that study. I'll be brief on ocean acidification because Ove gave a complete talk this morning about this subject, but briefly, the carbon dioxide emissions that are released in the atmosphere due to human activities are rapidly dissolved in the ocean, leading to a decrease in seawater pH. It is estimated that since the pre-industrial time, the seawater pH has already decreased by 0.1 unit. According to the Intergovernmental Panel climate change scenarios, we're expecting a further decrease of 0.3 units by the end of this century. And this is happening 100 times faster than ever observed in Earth's history. So the impact of ocean acidification on marine ecosystem is not well known, or there's a lot of studies on it. One of the most likely consequences of ocean acidification on marine ecosystem is the slower growth of calcifying organisms. So in fact, in seawater, 3 isoform or dissolved inorganic carbon are in equilibrium in the seawater, which are carbon dioxide, bicarbonate ions, and carbonate ions. And when carbon dioxide increases in the seawater, this provokes a decrease in carbonate ions. And this is a main concern because carbonate ions are the building blocks for calcifying organisms and biomineralization. And this raises a question, what would be the impact of ocean acidification on coral calcification? So a huge range of data have fueled this concern about impact of ocean acidification on corals. And all of them show that adult coral calcification will be highly affected by ocean acidification. But what about early life stages of corals? As you know, the early life stages in general are usually considered as to be the most vulnerable to changes in environmental conditions. And in corals, this step is really important for dispersal, renewal of population, and recruitment. As you can see on those pictures, these early life stages of corals are characterized by the onset of calcification, so the deposition of the first crystals of calcium carbonate, which will begin then the adult corals and the reefs that we can see. 3 main steps can be discerned in the process of onset of calcification. First, the deposition of the basal plate that you can see here in a ring, then the deposition of the septa, finally arrive at a more adult calcification, like a 3D architecture. So this table summarizes the impact of ocean acidification on the physiology of those early life stages of corals. And although ocean acidification doesn't have to have a major impact on survivorship and metamorphosis, those studies agree to say that skeletal growth and early calcification will be affected by this stress. My objective is to look at the molecular response of those coral juveniles to ocean acidification. In fact, one way to assess the effect of stress in general to organisms is to look at the molecular level. Organisms regulate a number of cellular processes, and the modulation and the regulation of gene expression is one of the most rapid and sensitive responses to environmental stress. And this gene expression profiling tool can provide us, it's a powerful tool to provide us insight into the mechanisms that are driving a decrease in calcification in corals. So for that purpose, we subjected early calcifying polyps of Acroporamylipora to different PCO2 conditions according to the IPCC scenario. So the first one is control. The second one, will be 750 parts per million of CO2, which correspond to moderate scenario according to the IPCC, and then 1,000 parts per million, which is like the pessimistic scenario. And we look at the short-term response of the early calcifying polyps to this. We use for that purpose next-generation sequencing, and more particularly the Illumina technique. So basically what you do is that you sequence the whole transcriptome of the early calcifying polyp in each of the conditions. This gives you a short sequence rate, and it's like a snapshot of gene expression in those different conditions. And by comparison with the transcriptome database that we have containing about 20,000 genes, we're able to look at the fold change expression of gene expression. So as you can see here, this technique is really good since the number of red corresponding to a particular gene will directly reflect the level of gene expression. Let's see at the preliminary result that we obtained. So this graph represents the number of genes differentially expressed between moderate PCO2 condition and control, but also between high PCO2 condition and control. We can see that we have about 5 to 10% of the genes that are differentially expressed between stress conditions and control conditions. And about 3% of them were common between the moderate and the high PCO2 conditions. I won't get into much detail here, but basically what we can observe between control moderate and high condition is 3 different patterns of expression. Either a gradual response to the stress, so either downregulation or upregulation. The second one is a lower threshold, so what I call lower threshold, so the gene responds between control and moderate, but not between moderate and high condition. And the last one is higher threshold, so the genes will go up or down at the highest PCO2 condition. How can we analyze the data? Sorry. As you can see, there's a mass of data. More than 2,000 genes are differentially expressed between those different conditions. Moreover, the mechanism driving calcification in corals are not well understood. So the next step here is the annotation of the database. So what does that mean? We need to assign a function of those genes by comparison to database existing in other organisms. And this is actually underway thanks to Sylvain Fauré. In the meantime, we had a quantitative gene approach focusing on genes already known to be involved in calcification and to see how those genes respond in acidified conditions. Just to remind you that coral skeleton is made of 2 main fractions. The first one is an organic fraction, which is mainly proteins, sugar, and also lipids. And this organic fraction is like a framework for the deposition of the mineral fraction, which is calcium and carbonate ions. So if you imagine here tissue coral, what you need to synthesize skeleton is first to synthesize the organic matrix molecules, and second to transport calcium and carbon from the external medium to the site of calcification. So several proteins, ion transporters, and also enzymes have been identified to date and also localized at the site of skeletogenesis, suggesting a role of those proteins in calcification. So as a candidate gene approach, we look at those genes already known, and I'll present you today only 3 of them. The first one is the carbonic anhydrase. So carbonic anhydrase is an enzyme involved in the carbon supply for calcification. As you can see on those pictures, this is hybridization, so Tracy showed you this technique, and this technique permits to localize in situ where the gene is expressed. So as you can see by comparison to this picture, the gene coding for this carbonic anhydrase in particular is expressed along the septa, so where the rate of calcification is higher at that stage, suggesting a role in calcification. In our experiment, these genes show gradually down-regulation under acidified condition, as you can see on this graph, representing the relative expression of genes in the 3 different conditions. So what I forgot to mention is that for this candidate gene approach, we validated or verified the fold expression of the fold change of gene expression, the Illumina sequencing data with another technique, real-time quantitative PCR, and which is a really sensitive method to quantify the gene expression. And as you can see, so in green quantitative PCR results and in red Illumina sequencing, the results are really similar, which is really encouraging and which validates the second gene that I'll show you is coding for the galaxin, which is an organic matrix protein involved in the nucleation of crystals. As for carbonic anhydrase, this gene mainly along the septa. And this gene, contrary to carbonic anhydrase, show a gradually upregulation under acidified conditions. So how can we explain that? This is maybe a kind of compensation from the coral to struggle with acidified condition, rather than in the other case, we can suggest that the carbon supply for calcification will be affected under acidified conditions. The last example that I show you today concerns 2 genes for which function is totally unknown. But if you look at their expression by in situ, they are expressed at the right time and at the right place for a role in calcification. So for example here, the expression is preferentially localized at the arboreal end of the metamorphosing larvae, so just before the larvae settle on the substrate. And here as well, corresponds to the basal plate of early post-settlement polyps. Those 2 genes are gradually downregulated under acidified conditions. So are they targets for new genes involved in calcification? This is another result that can confirm this hypothesis. So the next step here is to look at more candidates for genes involved in calcification, such as other carbonic anhydrase transporters, but also look at the genes that are responding the most to the stress, and to see what is the function of those genes and where are they localized in situ. But this candidate gene approach just gives you like an image of what can happen at a larger scale. But the main work here is to look at the functional classes of genes that are disturbed by elevated CO2. And we can do that after by the transcriptome annotation. One point that I would like to highlight, I talked today just about calcification, but metabolism might also be affected and will for sure be affected by ocean acidification. So it will be interesting to look at that as well. And 2 other experiments that could be interesting to do would be a long-term response. So in that case, we could be maybe able to look at the adaptive capacity of the coral to acidified condition, and also look at the combined effect of temperature and PCO2, as unfortunately corals are not only affected by one stress, but by multiple stress. Thank you, and I would like to thank Sylvain Fauré, Loretta Grasso, and David Miller for the transcriptomic part of this study, Eldon Ball and David Hayward for the in situ hybridization, and Andrew Baird, Min Chua, and Bill Legat for all what is related to the PCO2.